Hello guys, so I welcome you all to my new video. That video is about your phytoplasmas and spiroplasmas. So if we have to look at the phytoplasmas and spiroplasmas, they are also belong to a domain bacteria. Means they are a kind of bacteria, but they are having certain differences from your bacteria. So this we are all going to study in this particular video. So keep supporting me guys, keep subscribing, liking and sharing my videos so that I'll bring new and new topics for you to discuss. So let's get started. So if we have to talk about your phytoplasmas and spiroplasmas, you can say that phytoplasmas, they are having a kind of a pleomorphic shape. Pleomorphic shape means that will acquire a shape as per the organ in which the phytoplasma has entered, right? And your spiroplasmas, as the name is indicating, they are having a kind of a spiral shape, right? So major difference in terms of the shape among your phytoplasma and spiroplasma lies in the fact that phytoplasmas, they are pleomorphic, however your spiroplasmas are spiral, right? So now let's first discuss about your phytoplasmas, right? So phytoplasmas, as I already discussed, they are nothing but they are the bacteria. But how they are different from bacteria? They are different from bacteria with the fact that they are wall-less. Now here wall-less means that they do not have a peptidoglycan layer. All they can have is a unit membrane of cell, that is known as cell membrane, right? So they have a very thin cell membrane only. They are not having any kind of your murine layer, although they are your gram positive bacteria, so technically they should have that. But since they are different from bacteria, so they do not have any kind of your tough cell wall, so they are wall-less, they are pleomorphic, right? Generally having a smaller size, that is of 500 nanometers in diameter, and they are also having a small genome, that is 750 kbs around, right? And they are also a kind of pleomorphic, belong to your molecules, that too under your domain bacteria. And they also belongs to your gram positive bacteria because their ancestors were gram positive bacteria, so they should have the character of that. But because of certain changes, they do not have the wall structure like that of your bacteria, right? Now here you can see in the first picture that will show a healthy plant, however in the second picture that will show a one particular symptom of your phytoplasma, that is known as phyllodes, right? So here you can see a healthy one and a plant infected with phytoplasma. So we will discuss the different symptoms that are being caused by phytoplasma in the coming slides. Okay, now if you have to talk about your phytoplasmas, they require certain vector by which they can transmit from an diseased plant to a healthy plant, right? So this is a most common vector for the most of your phytoplasmas, that is known as your leaf hopper. Right, now let's discuss about the different symptoms that are being caused by your phytoplasmas. So among those symptoms, first is virescence. Virescence means a production of a excessive green color flowers. Second is phyllody. Phyllody means that in spite of having a normal floral parts, they get converted into the leafy structure, right? So that is your phyllody, right? Then third one we have is sterility. Now sterility basically means they will not have any kind of fertile condition and they will turn sterile. Then the next one symptom is witch's broom. Witch's broom means there is a proliferation of a shoot at a certain common point, so that is known as witch's broom. The next is elongation of your internodes, stunting, your root and leaf discoloration, leaf curling, etc. So these all are your symptoms that has been caused by your phytoplasmas. Okay, so most of the cases, or generally these diseases, they are detrimental, but in some cases if you are using the tolerant or the resistant varieties, so their damaging effect can be reversed as well to an extent. Okay, now here these are the pictures showing the different types of symptoms, be it your phyllody, virescence, witch's broom, etc. Okay, so these are the different symptoms that has been occurred due to phytoplasmas. Now here you can have a clear idea about its symptoms, like first picture showing a periwinkle plant which is healthy. Now in a healthy periwinkle plant, this is its flower. However, in a periwinkle plant which is affected by your phytoplasma, this flower, it is replaced by your leaf-like structures which is known as phyllody. Okay, second picture showing your healthy wheat as well as your wheat that is having the phytoplasmal disease known as dwarf. Then third picture is of a jujube tree. That's a healthy one, and that's a jujube tree which is infected with witch's broom. Similarly for your pylovenia, which is the healthy one, and this is your pylovenia which is affected with the phytoplasma and having the symptoms of known as witch's broom, right? So these are the different symptoms that has occurred due to the uh, effect of your phytoplasmas. Okay, now let's talk about the discovery. So these phytoplasmas were discovered in the year 1967 by a group of workers, and at the time of their discovery they are not named as phytoplasmas initially. They were at that time known as MLOs, that is the mycoplasma like organisms, right? So mycoplasma, they are known from a very long time as they are causing the diseases in animals, and also the mycoplasmas are the one which are causing diseases in animals and having a difference from their bacteria in the similar characteristics as we have for phytoplasmas. But at the time of their discovery, since they're causing the infection in the plants and having a somewhat similar characteristics to your mycoplasmas, they are at that time referred to as MLOs, that is your mycoplasma like organisms. Later it was in, I think, 1994, the name phytoplasmas was proposed to these MLOs, right? So in certain books you can also get this name MLOs, but now this name has been changed and it is referred to as phytoplasmas. Okay, so that is about phytoplasmas. Now this is the history that we are talking about. That is in 1967, and the Japanese scientists, they observed the microbes in the phloem of the plants having the yellow's disease. Now it is well known, whenever we are talking about yellow's disease, that means it is mostly associated with your phytoplasmas, right? So at in 1967 they observed that the phloem of the plants, they are infected with the yellow's disease, and the causal organism that was found associated with the disease, that is having a similar in morphology as to mycoplasmas. That's why at that time they are referred to as mycoplasma like organisms, right? Now these mycoplasma like organisms were later referred to as your phytoplasmas, because they are causing the diseases in plants, and this name was proposed in 1994, right? So now the difference that lies between your mycoplasma and phytoplasma is also that mycoplasmas are your non, or you can say to an extent they are culturable. However, your phytoplasmas, they are completely unculturable. That's why a name candidatus is also proposed with them, that we will discuss in the coming slide. Okay, so if we have to talk about the different species that we have of phytoplasmas nowadays, that is of around 900 species, or you can say 900 sequences has been deposited till now at NCBI gene bank. So right, and it is proposed that a status has been associated with your phytoplasma, that is known as candidatus. So whenever we are writing any name of any phytoplasma, it will always be like, for example, candidatus phytoplasma trifoli, a candidatus phytoplasma anything else, right? So these status candidatus was basically proposed with them, because here candidatus means that they are having a certain nutritional requirements, specific ones, and they're not cultivable, right? And if you have to identify the different species in a candidatus phytoplasma genera, you have to undergo the 16S RNA gene sequencing, and if there is a similarity of less than 97.5%, refers to a new species. If the similarity is more than 97.5%, it is considered to be the same species, right? So based upon those 900 sequences that we have deposited in NCBI, there are about 26 candidate species that has been described till now. Right now, as I already discussed, they are being vectored by the agent known as your leaf hoppers, because they have the property that they will replicate and multiply inside the gut or the tissues of your insect, particularly your leaf hoppers or plant hoppers. That's why they are important, right? So these genera are very closely related, like mycoplasma, spiroplasma, phytoplasma. So spiroplasma, you can say that is more closely related towards your mycoplasma, and your phytoplasma is somewhat distantly related from your mycoplasma. This you can remember in your mind, right? And basically we have to carry out the phylogenetic analysis, that is using the 16S rRNA sequencing to confirm that MLOs, they have a different clade as that of your spiroplasmas. Okay, so basically, as I already discussed, it was in 1994 the name phytoplasma was given, and that was proposed by your International Organization of Mycoplasmatology to refer them as phytoplasma, not as mycoplasma like organisms. Okay, now here you can see these spiral structures are your spiroplasmas, and these pleomorphic somewhat round structures are known as your phytoplasmas. The spiroplasma, that is more closely related to your mycoplasmas, and phytoplasmas, they are distantly related to your mycoplasmas. Although mycoplasmas are causing the diseases in animals and humans, however spiroplasma and phytoplasma are only associated with plants. Okay, now as we have discussed what are spiroplasmas, what are phytoplasmas, what are mycoplasmas, let's talk about the different diseases that has been caused by them. For example, in brinjal, the most important phytoplasmal disease is little leaf. Then in peach, another important disease is there, that is X disease. Then phyllidy was there, that is in sesame, which is the important one. Although all these are important, but your brinjal, little leaf, peach, X disease, then your sesame, phyllidy, which is broom, they are the most important one. And also your GSD, that was in grassy shoot disease, that is a phytoplasmal disease, and that was observed in sugarcane. It is also very important. And the yellow dwarf disease of rice, these are important. And then you can see the different areas, like little leaf, it is prevalent in all over the India. Similarly for your yellow dwarf and your grassy shoot disease, then your witch's broom and your phyllidy, they are mostly prevalent in all over India. So that will tell us about their importance. Okay, now if we have to look at the characters of your candidatus phytoplasma, now have you as you have understood about what are the characters of your phytoplasma, now how you can write them. So first we will be talking about its morphology. So as I already discussed, they do not have a cell wall, they will having only a single unit membrane, and they are pleomorphic, means they will not having a definite shape. Second is habitat. So they generally multiplies in the gut or the hemolymph of your insects, and that particular insect is your leafhopper, right? Then antibiotic sensitivity. Now as we know that since they do not have the cell wall, so they do not have any kind of sensitivity from your penicillin, because penicillin acts on the cell wall, but your tetracycline, which acts on the ribosome, they are sensitive to those. That is also a major difference between a bacteria and your spiroplasma and phytoplasma, that since in bacteria they are having a rigid cell wall, that is a murine layer or a peptidoglycan layer, they are more sensitive to your penicillin equally to tetracycline, because penicillin will act on the membrane or you can say the cell wall composition. However, your tetracycline will act on the ribosomes. But in case of phytoplasmas and spiroplasmas, since they have the ribosomes and other organelles as such, but they do not have the cell wall, so they are resistant to penicillin but sensitive to tetracycline. Right, now if we have to talk about their DNA based composition, so GC ratio, that should be around 23 to 29. Chromosomal size is comparatively less or smaller as compared to your bacteria. Their codon usage is also there, like we have three codons, UGG, UGA, UAG, but in this case your UGG will act as a stop codon, not for tryptophan. However, all these UGG, UG and UAG, they are the start codon, not the stop codon. But in case of your phytoplasma, this UGA, that will act as stop codon, right? Next is sterol in the membrane. Most of the bacteria will have sterols in their membranes, like cholesterol and others esters, but since they do not have any kind of that definite membrane, they do not have any kind of sterol, right? And ribosomal RNA, obviously they will have the ribosomal RNA, that will having the 16S and 23S as a two component of a RNA, right? So these are the important characters that you can keep in your mind regarding the description of any candidatus phytoplasma genera. Right, now here you can see this is particularly a leaf hopper, and here the red dots, they will depict basically how or you can say where this particular phytoplasma multiplies, and this arrow areas will represent its movement. Okay, so in leaf hoppers, generally they will acquire the phytoplasma from the phloem. We have two transmission, or you can say conducting tissues in plants, that is xylem and phloem. So xylem for water, and and phloem is for food or nutrients, right? So for your leaf hopper, it will acquire your phytoplasma from the phloem, and it get ingested, and it will move into the stylet or the food canal of your insect. From there it will be invaded into the intestinal tract, and then to the epithelial and muscle cells of the esophagus, where it multiplies and replicate. And when this particular insect, it will feed on another healthy plant, it will secrete that phytoplasma with the help of its salivary glands, and that helps in the transmission of this particular phytoplasma from a healthy to a disease plant. Okay, so this is how it will work. Now, if we have to talk about its classifications, I have already discussed it is based upon your 16S rRNA sequencing. We are generally concerned with that because that is a more conserved region, so it will have a better knowledge about its ancestry. And this 23S rRNA is a less conserved region, so it is not that much used. Now, if we have to look about its sequence similarity, so phytoplasma will have a 88 to 99% sequence similarity as that of your 16S rRNA. However, your eculoplasma, which is distant from your phytoplasma, that will having a different number, right? So here that is says why we are moving towards your 16S rRNA sequencing. Okay, now here you can see this is the new disease of phytoplasma that has been reported in carrot. Now this is not a phytoplasmal disease but a spiroplasmal disease that is caused by spiroplasma citri. As you can see, a kind of a these roots, or generally tap roots, they get infected in this case, and you can see the malformed carrot, because this particular phytoplasma spiroplasma, they cause the hormonal imbalance, right? So these are the result of that. Now the next question arises, obviously what are the differences can be there between spiroplasma and phytoplasma. So as already discussed, spiroplasma is more closely related to your mycoplasmas. However, your phytoplasmas, they are more closely related to your eculoplasmas, right? And the majority of your spiroplasmas, as they are associated with the insects, but your three spiroplasmas are your insect transmitted plant pathogens, that is Spiroplasma citri, Conchinelli and Phoenicum. These three, they are causing the diseases in plants. However, your all phytoplasmas, they are associated with plants as they are causing the diseases of plants only. However, your mycoplasmas, they are your human and animal pathogens. So this difference you can keep in your mind. I can repeat that mycoplasmas are your human and animal pathogens. Phytoplasma is a complete plant pathogen. However, in spiroplasmas, they are mostly insect pathogens, but three spiroplasmas are there. That was citri in citrus, causing the citrus to burn, Conchinelli causing the corn stunt, and Phoenicum that is also causing a phyllidy in ornamental plants. These are the three species of spiroplasma that are associated with causing the diseases in plants. Okay, so that is the major difference that you can remember. Now if we have to talk about the differences in mycoplasma and phytoplasma, it will goes like this. Like phytoplasmas, they are distantly related from the mycoplasmas. They are having a generally higher size, insect transmitted, and having the unit membrane. Okay. However, in case of mycoplasmas, they are having the sterols in their cell wall, which was not there in phytoplasmas. They can transmit themselves through different modes because they are mostly human and animal causing the human and animal diseases, ranges from 150 to 250 and they're generally a small bacteria that lack the cell walls, right? So this is all about it, guys. So if you have any query, you can please message, or you can also comment down in the comment section. Please like, subscribe and share. So the next disease that I'm going to discuss, or the next topic that I'm thinking of discussing in the next slide, will be about fruit diseases, or maybe on plant disease management. So you guys can comment down which topic I should cover, whether it is apple diseases or whether it is plant disease management. Okay, so thank you, have a nice day, and keep subscribing, liking and sharing. Thank you.